Ever stop and think about one of the deepest mysteries in all of biology? It's this incredible process that happens inside every living thing, a construction project so complex it's almost unimaginable, and it all starts from practically nothing.
So how on earth does that work? How does one single cell, this tiny, featureless sphere, have everything it needs, both the full blueprint and the entire construction crew, to build something as unbelievably intricate as a brain or a hand? I mean, it's one of the greatest puzzles in all of science, and we are finally starting to find the missing pieces.
So in this explainer, we're going to journey to the absolute cutting edge of science to see how we're starting to crack this code. We'll dive into the physics that literally shape us, the digital tools that let us see the hidden rules, and what all of this could mean for the future of engineering life itself. You know, for a long time, the simple answer seemed to be, it's all in our genes, right? DNA was the master blueprint. But it turns out that's really only part of the story. The real secret isn't just in the blueprint, it's in the physical forces that bring that blueprint to life. It's this amazing dynamic dance between our genes, chemistry, and, as we're learning, a whole lot of physics.
So let's start right there, with those physical forces. Because it turns out that our cells aren't just little bags of chemical soup. Oh no. They are incredibly sophisticated architectural structures, constantly pushing and pulling and sensing the world around them. And this brings us to this really cool concept from architecture, of all places, called tensegrity. Just picture a suspension bridge for a second, right? You've got the cables in constant tension and the big towers in compression. Well, our cells use that exact same principle. They build this internal skeleton that's both unbelievably strong and super responsive to its environment.
And this experiment just brilliantly shows how important those forces are. So researchers found that if you take a single stem cell and you put it on a really stiff surface, like a piece of glass, the physical tension literally tells it, hey, you should become a bone cell. But if you take that exact same cell and put it on a soft, squishy gel, it gets a totally different message. You should become a nerve cell. The cell's destiny, it's not just written in its DNA, but in the physical world that it actually touches. It's incredible.
This quote really highlights this massive shift in thinking that's going on. For decades, biology and physics, they were pretty much in separate worlds. But now, to solve this huge mystery of how we're built, scientists have to be fluent in both. The lines are totally blurring, and it's leading to these breakthroughs that were just impossible even a generation ago.
Okay, so we've got genes giving the instructions and physics providing the forces. But how in the world do you track the millions of interactions that are happening all at once inside a developing embryo? Well, for that, you have to turn to the digital world and build something called a digital twin.
There's this process called DevoGraph. It's pretty straightforward. First, you just film an embryo as it grows, cell by cell. Then, a program tracks every single one of those cells, turning each one into a point of data. And finally, it connects all those points, transforming that beautiful, messy dance of biology into a structured digital network an AI can understand. Now, to make sense of this massive digital network, scientists use a super powerful type of AI called a graph neural network, or a GNN. The easiest way to think about it is like mapping a social network. The AI doesn't see a blob of cells. Instead, every single cell becomes a person in that network, what the AI calls a node. And then all the connections between those cells, every physical touch, every chemical signal they send back and forth, those become the friendships and relationships linking all those people together. These are the edges of the network. So the AI's job is basically just to watch this entire cellular city evolve in real time. And by doing that, it can start to figure out the unwritten social rules, the fundamental algorithm that guides how this tiny community builds itself from the ground up. And these models are getting insanely powerful. For example, by analyzing the cellular network of a tiny worm called C. elegans, a GNN can accurately predict how its movement will change as it ages, from that energetic little wriggle it does as the larva to the slower, more deliberate motion of an old adult. It's actually learning the fundamental patterns of life. All right, this is where things get really mind-bending. Because once you start to actually understand the rules of development, the next logical step is to ask a pretty wild question. Can we start writing our own rules? Can we go from just observing life to actively programming it? This is the wild idea behind a brand new field called synthetic morphology. Now, the goal here isn't to go in and painstakingly edit a single gene, like in traditional genetic engineering. Nope. It's about becoming a cellular architect.
using the cell's own communication systems to guide them into building totally new structures all on their own.
And scientists have figured out there are three main communication channels they can use as control knobs. They can tweak the chemical signals cells use to chat with each other, they can change the physical forces we were just talking about, and they can even modify these subtle bioelectric fields that guide how tissues take shape.
You know, all this work trying to understand biological construction, it naturally leads to an even bigger question. If life, as we see it happening in an embryo, is basically a process of assembly with a clear set of rules, could it be that the very first life on Earth followed a similar path? Could understanding how life builds itself today tell us something really fundamental about how it all began?
The search for life's origin has a really long history. Way back in the 50s, the famous Miller-Urey experiment showed that life's basic building blocks could just form spontaneously. Then in the 80s, the RNA world hypothesis was huge. The idea that a single self-replicating molecule kicked the whole thing off. But some of the newer ideas are starting to sound really familiar. They suggest that life didn't start with a single magic molecule, but with a process. A new idea called assembly theory, for instance, argues that life's true signature isn't a specific chemical, but the measurable complexity of how molecules are put together, a construction project, but on a planetary scale.
And so, that leaves us with a truly profound thought. As we master the rules of biological construction, from the physics of one cell to the digital logic of an entire embryo, a whole new possibility starts to come into focus. Are we just learning to read life's ancient blueprints, or are we on the verge of becoming the architects of life ourselves?
Welcome to the Deep Dive. Today, well, we're really covering some ground. It's a massive intellectual journey we're starting.
It really is.
Our sources bridge fundamental physics and engineering think cables, tension, stiffness, all the way to cutting-edge deep learning.
Yeah, it's quite the range.
We're tackling this really interdisciplinary question. How does the physical world actually dictate biological development? And how can computation even begin to map that complexity?
It's an incredible stack of material. You've got classical mechanics rubbing shoulders with, modern cell biology and network science.
So what's the goal for you listening?
Well, our goal is pretty simple. Give you the ultimate shortcut to the key aha moments right at that intersection. We're going to zero in on some really powerful concepts. Things like tensegrity in cells, the counterintuitive power of just how squishy something is, the substrate, and even the subtle physics of things like topological symmetry breaking.
Okay, let's unpack that. Because when we talk about the physical world shaping life, we really have to start with the cell itself, the mechanics.
Mm-hmm.
You might picture a cell as just, a soft blob, right? A fluid-filled sac.
That's the common image.
But researchers are increasingly modeling the cytoskeleton, the internal scaffolding, using ideas straight out of architecture. Engineering, really. We're talking about tensegrity.
That's the word, tensegrity. It's short for tension and integrity, and it's actually a perfect model here.
And how so?
Well, it describes structures where elements under compression, like rigid bars or struts, they're held separate. They're stabilized only by continuous elements under tension. Think wires or strings. It's pre-stressed.
Like a geodesic dome, or those sculptures where rods just float.
Exactly like that. Those are great visuals. The rods seem to float, held only by tension wires. That's tensegrity.
Okay, so applied to a cell, the struts are like microtubules, actin bundles.
Precisely. And the tension elements are the stress fibers pulling everything taut. And the sources show how even a simple, three-strut tensegrity model can act like a cell. It changes height, it twists, it transmits forces depending on what's touching its surface.
So that internal tension gives the cell its shape, but also how it senses the outside world.
That's the key. It defines how it detects and responds to external forces.
And speaking of the outside world, this brings us to, I think, a really huge insight for you listening. It fundamentally reframes development.
What's that?
The substrate dictates fate, the surface it's sitting on.
Ah, yes. This is huge.
We tend to think stem cell differentiation is all chemical, right? A cocktail of growth factors.
The standard view, yeah. Add this, get that.
But the mechanical environment outside the cell, how stiff or soft the extracellular matrix is, or whatever it's growing on, that profoundly influences its fate.
It's so counterintuitive, isn't it? Why should the squishiness of the petri dish matter more than the chemicals in the broth?
Exactly.
But the studies are clear. Elasticity is often the primary driver. You put cells on really soft surfaces, low elasticity, you often get nerve tissue. Medium elasticity surfaces, they tend to push stem cells towards muscle.
And the stiffest.
The stiffest, high elasticity surfaces, They reliably make bone tissue. It's like a physical dial controlling a biological choice.
That is wild. A mechanical command just overriding chemical signals.
It seems so, in many cases.
And it's not just stiffness, is it? We also saw research on physical patterns.
Oh, yeah, the hydrophobic and hydrophilic squares.
Right. Cells behave completely differently based on the size and spacing of those squares on the dish.
The geometry, the texture of their tiny worlds.
Yeah.
It's constantly feeding back into what the cell decides to do.
Which leads us to, well, a point of frustration that came up again and again in the sources.
The data gap.
The data gap in biology. We know mechanics matter, but getting the right kind of data is tough.
It really is. Cell biologists need fundamental engineering data. You know, proper stress strain curves, force distance curves for specific cell types, standardized stuff.
But that requires...
It requires the rigor of mechanical quantification. It means talking to mechanical engineers.
And that's where the problem lies, the communication.
Often, yeah. You have these two expert groups, but they don't always speak the same technical language. The engineer asks for Young's modulus.
And the biologist says,
It's stiff or it's soft, you know. So getting that standardized fundamental data needed for truly predictive mechanical models, it's slow going because of this cross-disciplinary barrier.
Okay, let's shift gears slightly. Life and death. Because this finding on apoptosis, programmed cell death, connects mechanics and cell fate in a really shocking way.
Oh, this is fascinating. We know apoptosis happens, but what happens next is key.
Compensatory proliferation. Neighbors dividing to fill the gap.
Right. But it's not random which neighbors divide. The pattern is very specific. It's defined by, get this, inhomogeneous mechanotransduction.
Okay, break that down. Mechanotransduction is...
Just the cell sensing and responding to physical forces, mechanical forces.
And inhomogeneous means it's uneven. It varies.
Exactly. The sensing isn't uniform across the tissue or even across a single cell sometimes. So after a cell dies, its neighbors feel that change. They sense the gap, the change in physical stress.
And that feeling, that mechanical stimulus, is the trigger.
It acts as the selection criterion. It's incredibly precise. Only the neighboring cells that show both a large nucleus and feel a large mechanical stimulus get picked to divide.
Wow. Not just one or the other, both.
Both. Large nucleus, large mechanical signal, those are the cells selected to divide and fill the space efficiently. It's a localized, mechanically driven selection for tissue repair.
That's incredible. Physical sensing literally decides who gets to reproduce.
It really drives it home, doesn't it?
Okay, so scaling up now. We go from single cells making decisions based on mechanics to tracking, well, an entire organism developing. Thousands of cells moving, dividing, changing over time.
Now we're talking 4D, 3 dimensions of space plus time.
And that kind of complexity, tracking all those moving parts and their relationships. It demands a shift from just mechanics to computational networks.
Absolutely. Managing that sheer volume of data, the constantly shifting connections between cells, standard tools, even typical neural networks that are good with features, they kind of fall short.
Why is that?
Because you need to model the relationships, the connectivity. And that's where graph neural networks, GNNs, come in.
GNNs. Why are they suited for this?
Because they're built to model interactions. Instead of seeing a cell just as an object with properties, a GNN treats the whole tissue or organism as a graph. Cells are nodes, connections are edges.
So it's about the network structure.
Exactly. That structure is perfect for tracking things like, say, the locomotion of an aging C. elegans worm. The sources mention modeling the worm as a chain, right?
Yeah.
And the GNN captures the complex changing forces between adjacent body segments. It's not just analyzing static data, it's modeling a dynamic, interconnected system.
So GNNs map the changing connections. And the ultimate goal of all this modeling, is it the digital twin?
That's the holy grail, in a way. The courses talk about a connectome-based digital twin of C. elegans that can actually show sensory motor behavior.
So not just a picture, but a functional model.
A highly functional model.
Huh.
It incorporates the worm's full neural wiring diagram, the connectome, and crucially, it can predict movement based on what the virtual worm senses.
Like predicting how it moves towards food.
Precisely. They modeled chemotaxis, the worm seeking a chemical gradient, like food using a radial Gaussian distribution. And the digital twin's predicted movement matched reality. It's a functioning virtual replica.
A prediction machine built from the ground up.
You know, it's interesting how these high-level models sometimes highlight really basic critical factors we might otherwise miss.
Like the cell density example.
Exactly. The lesson from cell culture about density. It seems obvious density affects growth rate, right?
That's the standard assumption. More cells, faster growth, maybe.
But the data shows something more fundamental. Density is critical for survival, just staying alive. Yeah, this is a vital takeaway for anyone doing tissue culture. Cell populations starting too sparse, the number cited was below 1,700 cells per square centimeter.
What happens to them?
They approach extinction within about 6 days. They just die off.
Extinction. Not just slow growth, but gone.
Gone. They literally can't make it. They need a critical mass of neighbors, it seems, to establish the right kind of collective signaling, like a quorum sensing maybe, a community support system just to survive being seeded.
But above that threshold.
Above 1,700 cells per square centimeter, they grow fine. They head towards confluence. It really puts collective action front and center for initial survival, not just individual cell health.
Okay, let's zoom out again.
Yeah.
To the big picture of morphogenesis, the process of forming shapes and development. What physical forces are really driving this?
Well, one critical factor researchers are isolating is the interplay of curvature and flow.
Curvature of the tissue itself.
Yes. The collective behavior of cells, how they move together, is strongly affected by the curvature of the sheet or tube they form, sometimes regardless of other factors.
There was a Drosophila embryo example, right?
A great one. The embryo undergoes dramatic shape changes driven by polarized tissue flow. Cells are moving in coordinated ways.
And what drives that flow?
It turns out it's driven by a geometric coupling. It's the interaction between the cell's internal contraction machinery, the actomyosin network at their apical surface, and the overall curvature of the tissue they're part of.
Geometry is coupling to the internal motor.
You got it. And here's the really surprising part the sources highlighted.
What's that?
This flow, this shape change, seems to happen independently of how strongly the cells are sticking to each other, independent of adhesion.
Wait, really? You'd think they need to be glued together to flow as one.
That's the intuition, right? But here, it seems the geometric constraint, the curve of the tissue, is the dominant organizing force. It overrides the need for super strong cell adhesion to get that large-scale movement. Geometry rules.
That reliance on geometry, on packing and constraints, it connects to another concept from physics, doesn't it? The jamming transition.
Jamming, yes. This comes from soft matter physics.
Explain that. What is a jamming transition?
Okay, picture particles, or in our case, cells in a dense environment. They can shift from a state where they can flow past each other, like a liquid or a fluid.
Mobile.
Right, mobile, to suddenly, rapidly becoming immobile, locked up like a solid, think of cars on a highway. Below a certain density, traffic flows. Hit that critical density.
Gridlock. Everything stops.
Instantly.
Yeah.
That sudden switch from flowing to frozen, that's the jamming transition. It happens at a critical density.
And we know cells and tissues can undergo this. They stop flowing, act more solid.
Yes, it's observed in developing tissues, wound healing, even cancer metastasis. But what gets really interesting is when this jamming happens on a curved surface, not a flat one.
Like inside an embryo, what difference does the curve make?
It introduces geometric frustration and nonlinearity. Basically, the curvature forces the cells into arrangements they just can't achieve on a flat plane. It's harder to pack them perfectly.
So you get defects, gaps.
Exactly. You get structural defects, specific rearrangement patterns, maybe stress concentrations. The shape of the container fundamentally changes how the jamming happens and what the final locked-up state looks like.
It makes sense. The physical world, the container, is always imposing rules.
Yeah.
Okay, let's push this right to the edge now. The origin of life. How could metabolism even start?
Yeah. The sources looked at simulations of early life vesicles, trying to see how simple autocatalysis, like basic metabolism, could spontaneously get going inside these primitive compartments.
What was the big hurdle they found in the simulations?
Leakage, a massive leakage problem. These simulated early vesicles were just too permeable. Molecules leaked out incredibly fast.
So even if reactions started inside,
The products and reactants would just diffuse away before they could build up to the concentrations needed to sustain anything lifelike. The system couldn't accumulate complexity. Leakage always won.
so how did they solve it in the models? What made it work?
The breakthrough came when they added what they called a memory component.
Memory. In what sense?
It was some kind of intermittent physical mechanism. something that temporarily slowed down the leakage, maybe a brief period of cooling that made the membrane less permeable, or maybe the absorption of some large molecules that clogged the pores for a bit.
And that temporary slowdown was enough.
It was enough to prevent everything from leaking out. It allowed the vesicle to retain a small fraction of its contents from the previous cycle, the previous day in the simulation.
Ah, so it could carry over some progress.
Exactly. It could incrementally build up the necessary molecules over time, cycle after cycle, eventually reaching the threshold needed for sustained reactions. The system needed that little bit of physical memory to overcome the constant drain of leakage.
That's a beautiful idea. Physical memory enabling the transition to life. So, thinking about defining life itself, there's this framework called assembly theory.
Right. Assembly theory offers a different kind of definition. It argues that life is unique because it's the only kind of physics we know that generates highly complex objects, things that couldn't just spontaneously assemble.
And how do they measure that complexity?
Not just by the chemical formula, they use something called the assembly index. It's basically the minimum number of steps or bonds needed to construct that object from its basic constituents.
So more steps means higher assembly index.
Precisely. And really complex objects, things with a high assembly index, requiring a long, specific construction history that's a strong indicator they were produced by a living process. It's a quantitative measure of generated complexity.
And we can see this emergence of function, even in simple non-living systems. There was an analogy.
The toy model analogy, yeah. It was meant to illustrate functional rescue. They described a non-viable vehicle, basically, some assembled construct that was asymmetrical and didn't work, couldn't perform its function.
Okay, a failed machine.
Right. But then this completely non-functional thing could be instantly rescued, made functional, just by adding one specific component, like adding a tiny rotor.
And suddenly it works.
Suddenly it works. It shows that clear transition, crossing a functional threshold, maybe analogous to non-living, to living, just by achieving a specific, complex arrangement of parts. Function emerges from structure.
Wow, what a journey today. We started down at the level of tensegrity basic mechanics defining cell shape.
Tension and integrity, yeah.
Then we scaled up through the abstract logic of graph neural networks, tracking thousands of cells moving in 4D.
Mapping the network.
And we ended with these fundamental physical principles, jamming transitions, the crucial role of curvature sculpting the embryo, and even that need for physical memory at the dawn of life.
And along the way, seeing how the mechanical environment is literally fate for a stem cell.
Right, and how collective density is just basic survival for a new culture.
Absolutely.
If we try to tie this all together, it seems to raise some deep, almost philosophical questions, doesn't it? Especially about these complex models.
It really does. There's a fascinating debate kind of inherent in modeling development this way. As observers, we instinctively assume development works towards a goal, right?
We assume the embryo wants to become the adult. It's teleological thinking, purpose-driven.
Exactly. But does the embryo itself have a goal state? Or is it maybe just following a sequence of deterministic local rules, physical rules, chemical rules, where the final adult form is simply an outcome, a consequence, not a predefined target it's aiming for?
So the model suggests maybe the embryo doesn't have the normal adult as its goal state.
That's what some sources suggest, yeah. It fundamentally challenges how we frame development, function, even purpose in biology.
That is a stunning reframing.
Yeah.
It almost makes you wonder how development is even reliable if it's just local rules. How does it not just go off the rails?
Well, that's where constraints come back in. That might be the key. You should explore this idea called phenotypic concentration of dimension.
Phenotypic concentration of dimension. What's that?
It's the idea that even though organisms have this, near infinite genetic and cellular complexity, the actual variety in how they physically develop their phenotypic variability can often be captured in a surprisingly low dimensional space.
Yeah. Meaning, even with all the complexity, the possible outcomes are limited, funneled.
Exactly. The vast complexity seems to be constrained, channeled into a much narrower, more predictable developmental path. The complexity gets concentrated.
The complexity is constrained. It's the architecture to the constraint itself that defines the outcome. Fascinating stuff.
Really makes you think, doesn't it?